Very good morning to all of you. First of all, I would like to thank the organizers and the co-organizers, especially Arijit for inviting me here because I had a long-drawn intention to visit Math Science. So this is the first time I am visiting Math Science. Thanks, Arijit. So to start with, I would like to draw your attention towards the word non-coding because our lab is basically interested to look into the modulatory aspects of non-coding RNAs on the protein coding genes. So as the title of my talk goes, we are actually looking into the coding and the non-coding RNA interactions and how this interaction is playing a big role in disease biology. To start with, I would just talk about the main research themes which are going on in our lab. So broadly, we are working on different types of cancer. Second, it is the stem cell-based regenerative therapy. And the third one is on different neurodegenerative diseases. So as I have told that we are interested to look into the interactions between the coding and the non-coding RNAs. And we are incorporating machine learning-based approach. Now, today I shall be mainly talking about acute myeloid leukemia system for the cancer part which we are working. So here basically we are trying to develop a non-invasive mode of preliminary detection of acute myeloid leukemia, which is involving this coding and non-coding RNA interactions. So this work has already been published and it is already available in GitHub. The second one is this stem cell-based regenerative therapy, where we are trying to predict mainly the safety and the efficacy of the newly generated induced pluripotent stem cell-derived cells, which can be used for regenerative therapy. And a very new aspect which we have started in our lab is perhaps looking into the risk of Alzheimer's disease in elderly people, whether we can predict the risk of AD in elderly people. So today I shall be sharing with you two stories. One is regarding this AML and the second one is regarding this stem cell-based regenerative therapy. Now before I go ahead, I would just take you shortly through the non-coding RNA, a very brief overview about the non-coding RNA world. So initially, if you look at the human genome, you will find that the major part of the genome was a dark matter previously. A very little portion of it codes for protein. Gradually, we came up with that this huge portion of the non-coding RNAs, they are not actually the dark matter, but it matters a lot. We find that these non-coding RNAs, they plays a major role in stem cell maintenance, differentiation, and different types of diseases, including cancer, as well as it is also guiding the stemness of the cancer stem cells, and lot other important aspects are there. So if you look at the different types of non-coding RNAs based on their length, there are the small non-coding RNAs and the long non-coding RNAs. Among the small ones, we know that this is a very well-known one, that is the microRNA, siRNA, and the less known one is the PV interacting RNA and the piRNAs. And we all know about this long non-coding RNA. So in my lab, we work with microRNA, piRNA, as well as long non-coding RNA. But today I shall be focusing on microRNA, the regulatory aspect of microRNA in these disease systems. So now let us start with the acute myeloid leukemia system. So what happens here is, basically it is a malignant tumor and we all know that it's sort of a blood cancer where there is a neoplastic proliferation of the myeloid cells. And here basically there is an accumulation of the immature myeloid cells which are called the blast. So you can see here these are the blast population within the bone marrow samples. And the gold standard test which is done is basically to take the bone marrow aspirate from the patient, which is a very invasive procedure, and then go ahead towards testing for the disease. So we are next wanted to see that how basically the bone marrow and the peripheral blood biology differs in case of an AML patient. So we looked upon the literatures and we found that this particular mutation in the FLT3 gene is very detrimental, and this is basically responsible for the relapsing of this AML disease, and this can be tested from the peripheral blood. Next, this Winkup et al. He performed a very comprehensive analysis towards looking into the morphological and cytochemical analysis of both peripheral blood and bone marrow samples from different patients. And he found that peripheral blood samples, it also can be used for the diagnosis of this AML. So with all these informations, we wanted to look closely what are the different common regulatory mechanisms which are present within this peripheral blood and bone marrow of AML patient that leads to the disease etiology. Next comes down to the importance of microRNA as a key player in this disease. So here, as I have told you, that microRNA is involved in different diseases, including different types of hematological malignancies. And as I mentioned, that this is a very important, FLT3 mutation is a very important mutation for AML detection. So here also, now around 55 is upregulated and is associated with the different increased myeloproliferation. So next, this is another 29B, now 29B1, this is another tumor suppressor microRNA, which is also found to be involved in case of the AML patients. So next, what we thought that if we can incorporate the microRNA regulatory aspects and whether we can precisely design the AML diagnosis panel by focusing towards the role of microRNA in regulating the entire transcriptome of the AML patient. So with all these things on board, we designed our workflow. So here what we did was we looked into the gene expression data of the AML patients which are coming from bone marrow as well as from peripheral blood. We next considered patient tissue samples as well as the cell line also. So KG1 is the cell line for the AML bone marrow, and here HR60 is the cell line for the peripheral blood of the AML patient. So we considered both the tissue samples as well as the cell line samples. We collected the microarray as well as the RNA-seq data. So this we did for both the microRNA as well as for the genes. And we did the differential gene expression analysis. Finally, we brought out the microRNA target gene enriched profile, we filtered it out, and we screened them based on their importance for AML biology. And finally, we got the final set of microRNA target genes, which has been used as the feature set to design our prediction model. So now if we move on towards the data set, so this is the microarray data which we have taken from GEO database. So this is for the mRNA genes, that means the protein coding genes, and this is for the small RNA. And these are the RNA-seq data sets from GEO which are obtained for the long RNA-seq data, that means for the mRNA as well as the microRNAs. So once we did the analysis for the differentially expressed mRNAs, you can see here this is the result for the RNA-seq data. If you look at the clustering profile, you will see that all these samples, these are from the bone marrow, either from the tissue or from the cell line, they have clustered together. And these are the control data sets. So the control data set are the CD34 positive cells from the normal bone marrow and peripheral blood. So they are clustered together. The same result has been obtained for the microarray data analysis also. So with these screen set of the differentially expressed mRNAs, we next moved on towards looking into their interaction with the microRNAs. And here we tried to look into the functional analysis of the microRNA target genes which are present within this particular differentially expressed mRNA gene set. So here you can see that all of these functions, these are very much enriched functions which are involved in the AML biology. So here also these are the enriched pathways which we have found here. So next, if we look at the entire overview of the different types of functions and the pathways which are being regulated by the microRNAs here. These are the cellular differentiation, hematopoiesis, migration, adhesion, proliferation, apoptosis, cell survival. So here you can see this red map are the microRNAs and these are their target sets. So among these, we tried to check whether how many of them have already been reported in literature. So this is one of the microRNA genes whose expression is high already in AML patient, which has already been reported previously. So with these sets, we tried to develop the prediction model. So here we have adopted both the UMAP, that is the unsupervised clustering, UMAP unsupervised clustering technique, as well as the logistic regression-based supervised learning strategy. And we have developed what is the name of our model is TB-AML Pred, which predicts AML from the patient's peripheral blood sample. So it, on one side, eliminates the need of the invasive bone marrow sample collection. So going into the detail of this model, so here the training data basically consists of the gene expression data which we have obtained from the bone marrow and as well as the KG1A, and the validation data is from the peripheral blood and HL60. So here you can see, that the red and the green ones, these are basically the bone marrow and the peripheral blood contrails. So they have clustered together, and the blue and the orange ones, these are basically the disease set. So the bone marrow AML and the peripheral blood AML, they have clustered together. So this is the UMAP visualization. And next, when we have tried some unknown data set on it, here we find that the control is the red ones and the AML and the validation AML, that is the green and the blue ones, they have clustered together. So when we look at the scores, you can see here if it is greater than 0.5, then it is AML, and if it is less than 0.5, it is normal. So here you can see all these samples, they are greater than 0.5. So they have been detected as the AML and are rightly clustered with the AML population. So looking at the unknown RNA-seq data, here also we have found that this CD34, this control set, the red one is separated out and the validation AML, they have separated out. So to get a proper score of this, here you can see that all of them have been detected correctly as the AML. So if we go towards the working arm where you can use it, here basically you can use either the microarray data or the RNA-seq data for the unknown sample, and then it is going to analyze it. So it will use read counts if it is from the RNA-seq data, and it is going to use the normalized intensity values if it is from the microarray data, and finally you will have the proper clustering to provide the status of the sample. So to conclude this first work, here basically what we have found is the microRNA mRNA target pairs, they are basically playing an important role irrespective of their sample source being peripheral blood or bone marrow, which is providing a clue towards how the disease is progressing or towards detecting the disease. And next is we have tried to develop a non-invasive procedure of preliminary AML detection, which can avoid the collection of sample from the bone marrow. So this work has been published in Computational Biology and Chemistry. You can take a look at it for detailed technical details of it. And here in the GitHub, you can find this model. You can use it there. So coming on to my second story regarding the induced pluripotent stem cell derivatives, I will just quickly go through a brief overview of what are these pluripotent stem cells. So these pluripotent stem cells basically have the potency to give rise to different types of cells within our body. So that is why it has brought the importance of regenerative therapy. So initially the work started with the gold standard embryonic stem cells, which comes across the development of an embryo. But later on there were certain ethical concerns to use them for regenerative therapy. And that is why Shinya Yamanaka and James Thompson, they came up with the concept of developing induced pluripotent stem cells by using a cocktail of transcription factors. We can actually use the differentiated individuals and push them up the epigenetic landscape to give rise to induced pluripotent stem cells. So next, we thought that, the strange mystery here is these induced pluripotent stem cells, they are behaving like this classic character of Dr. Jekyll and Mr. Hyde, because Dr. Jekyll, we know that he was a noble doctor, and Mr. Hyde was a criminal who was the other part of the same person. So here we see that when we are using this iPSCs, it has brought a tumorigenic potential within it. On one side, it has got a regenerative potential, and on the other side, it has brought a tumorigenic potential. And here also, once again, microRNAs are one of the key players towards inducing such tumorigenic potential within these iPSCs that has been reported already. So with this thing, we next thought that if we want to eradicate this thing, if we can differentiate the cells towards a lineage of cells what we want, desired lineage, then whether we can avoid this problem of oncogenicity. So here also the same question arises that whether this differentiated population, they are truly devoid of oncogenic potential. So next came the problem of dissecting the oncogenic potential of stem cell derivatives. So I did a very preliminary work towards the year 2012, where I found that within these stem cell derived cells, I analyzed the transcriptomic data and found that there were certain inherent oncogenic properties even within these stem cell derivatives, which was already published there. So next, we want to come up with these questions, because we know that these induced pluripotent stem cells, they give rise to cells corresponding to the three germ layers, what we call ectoderm, endoderm, and mesoderm. So all the cells belongs to these three germ layers. So that is why we wanted to see that whether these oncogenic signatures retained by these iPSC derivatives, they are present within or the corresponds to all the three germ layers. Next is, does microRNA contributes towards such oncogenicity? The third one is whether the reprogramming method that plays a role towards imparting such oncogenicity within these iPSC-derived cells. The reprogramming method means when you are developing an iPSC using a cocktail of transcription factors, you will see that there are different factors like c-MYC, L-MYC. So these are oncogenic basically. So whether these reprogramming factors are inducing such oncogenicity within this stem cell derivative, that is also one thing that should be checked. Last one is, with all these informations on board, can we predict the oncogenicity status of a newly generated iPSC derivative? So this we thought that somewhat it will be useful for a lab who is newly generating an iPSC derivative. They can check their cells and they can see that, oh yes, it is okay to go for regenerative therapy, or no, it is not okay to go for regenerative therapy. So these were the questions we had, and next we started on to design the experimental workflow. So on the first phase, we studied the cells corresponding to the three germ layers. So what we did is we collected the gene expression data sets for the iPSC-derived cells corresponding to these three germ layers, ectoderm, endoderm, and mesoderm. And we also collected the microRNA expression data corresponding to these cells. We again sorted out the interactions among them, and then we did this target prediction of these microRNA target genes. And in the second phase, we worked on the iPSC reprogramming methods, where we collected the gene expression data of iPSC-derived cells, where the iPSCs has been generated by all these different reprogramming methods. And we mapped these for phase 2 and phase 1 data and came up with a set of microRNA target enriched features, which can be used to develop a prediction model, which finally can help to predict the status of the newly generated iPSC-derived cell. We are also validating a part of this work because we are taking up or picking up certain microRNA, mRNA targets from their feature set, and we are validating it in our lab also. So now this is the experimental grouping what we did. So, it was very difficult to sort out the expression data because we had to select the cell types for each of these germ layers for whom we got the expression data, because we needed to have both the microRNA as well as the gene expression data for that particular cell types. So these are the three tissue cell types which we selected. So our control group was the primary cell type. So here it is the primary neuron, primary hepatocyte, primary cardiomyocyte. And for the first test samples, it was the iPSC-derived neuron, iPSC-derived hepatocyte, and iPSC-derived cardiomyocyte. For the negative control group, actually, we had the carcinoma, that is the neuroblastoma corresponding to these neurons, hepatocellular carcinoma corresponding to hepatocyte, and sarcoma corresponding to cardiomyocyte. So what we wanted to see is that if this has properly been differentiated individual, the gene expression should actually cluster with their primary cell. If not, let's see what happens. So that is what we wanted to check. So here what we did was we collected the gene and the microRNA expression profiles for all the samples which we obtained from the GEO database. So when we looked at the clustering of the cells, gene expression data, what we found here is, you see the iPSC-derived neuron, which should ideally cluster with primary neuron, it is clustering with neuroblastoma, the cancerous counterpart. Again, here also when we look at the microRNA expression profile, the primary neuron is clustering with the iPSC-derived neurons. The iPSC-derived neuron is clustering with the neuroblastoma, but the primary neuron is separated out. So when we look at the expressions for the microRNA as well as the gene expression data for all the different other germ layers, the endoderm and mesoderm, here we also got the same type of clustering. So from here we concluded that the iPSC-derived cells, they are closest to their cancerous counterpart irrespective of the germ layers. So now the work started on towards fishing out the important set of genes which are actually responsible for this type of clustering information. What we did was, we once again predicted the target genes, microRNA target genes for all the 3 germ layers, and we next analyzed the functions of these set of genes which are actually playing a big role here. So finally we came up with 116 pathways which are common across all the 3 germ layers, because we wanted to find out a common signature, common oncogenic signature which is pertinent or which is present across all the 3 germ layers irrespective of the germ layers. So this is what are the common pathways we found among the 3 germ layers. So next we wanted to look into the effect of reprogramming method. So here, as I have told you, that these are the different types of transcription factors which are used for these different types of reprogramming methods. And recently reports have also come up, although we have mentioned here that it is a non-carcinogenic method, but still Lin-28 is, or even with Lin-28 there are certain carcinogenic effects coming up. So recent reports are there. So what we did was, here we wanted to see that whether oncogenicity is induced in iPSC derivatives irrespective of the method of its generation. So here we can only get data set for iPSC derived endothelial cell, because we wanted to have these data for iPSCs which are generated by all those reprogramming methods. And here the primary counterpart is the human umbilical vein and endothelial cell HUVEC, and the cancerous counterpart are the breast cancer and the ovarian cancer. So here when we did the clustering, we find here that this HUVEC is separated out, and this iPSC-derived endothelial cell, it is clustered with the cancerous counterpart. And this is happening for all the data sets coming from the different reprogramming methods. So here once again we looked into the set of the genes and we fished out the pathways. Here we find that the 69 pathways are common across all these reprogramming methods. So next we wanted to overlap phase 1 and phase 2 analysis and come up with a clean set of microRNA target gene regulated pathways which are, very much responsible for the oncogenic perturbation, as well as which will show that irrespective of the reprogramming method as well as the germ layers, these are actually inducing the oncogenicity within these cells. So if you look at the pathways here, these are the microRNA target gene associated pathways, and with this set of information we try to build the prediction model. So here basically once again we have used supervised UMAP clustering as well as the logistic regression based information. So in this way we have designed the prediction model. So now if I show you the result here, you see here first for the training data, the primary hepatocytes, and there is the hepatocellular carcinoma HCC. So here you can see that the primary hepatocyte, which are the green samples, and the unknown iPSC-derived hepatocytes are these black dots. The HCC are the red dots. So here you can see the clustering is very different. If you look at the score here also, it will give you a very good impression. So if it is 0, it will be closer to normal. If it is 1, it is closer to cancer. So you see here, the different data sets are behaving in a different way. So some are 1, some are 0. So those which are 0, they are closer to normal, and those which are 1, they are closer to cancer. So we wanted to take a look into this data set, that why this variation is happening, where lies the problem with those which are not actually 0. It should ideally be 0 because it should be devoid of oncogenic potential. So we found that the source cell, which have been used from 2 different persons for generating the iPSCs, that might have induced this oncogenicity within this particular iPSC-derived hepatocyte. Similarly, we looked into another data set, that is the primary neurons. So here we find that these 2 data, they are giving a result of 1. So here also we check that the source cell, they are different for iPSC generation. So that might lead towards this spurious oncogenicity within this population. And for the third one here, all have been shown 0. So they are properly clustered with their primary counterparts. So we can say that they have been properly, differentiated. So here comes the prediction model. So here basically you need to have the microarray data from the iPSC derived sample. And finally, you are going to get the status of the iPSC derivative in the form as either 0 or 1. And what we will additionally provide is the set of the important set of top 5 genes which are actually making it and shifting the cells towards this oncogenicity. So this is going to direct the wet lab people to some extent to show that these are the problems which are there within the the profile of these iPSC derived cells. So if you look at the entire biology and the whole thing, you will see that the major players here, what we found is this MOR148A3P, which is actually have a reported role towards promoting tumor angiogenesis. It is targeting this particular gene, which is downregulated here. And this downregulated gene actually inhibits tumor growth and metastasis. So this is one axis. The other axis is this MOR182.5P. So this particular, it also has a reported role which suppresses cell apoptosis, and it regulates this ROQ gene, which again regulates EMT via phosphorylation. This axis again, once again, has a role in chemotherapy, so chemoresistance, and this one is another one is the MORFIL11.5P. So here it basically is downregulated, is a tumor suppressor microRNA. It controls proliferation. So this particular target gene controls proliferation, migration, and invasion, which actually is upregulated in our system. So we have been able to validate this axis, this MOR148A3P and this target gene in our lab. So the rest of the validation work for this and this are going on in our lab. So hopefully we will be finishing it up within 1 month and communicating this paper. So lastly, I would like to acknowledge the funding agencies. And here is my lab network. So Origita, Troy, and Gulrod, they worked for the, with the acute myeloid leukemia system. And here Bapti, Origita, and Gulrod has worked for the second problem which I discussed with you. So here is my entire lab at Bose Institute. And to end, networking is the mantra to success so that we can have fun doing research. Thank you.
So hi, Jura, very nice talk. I have 2 questions. One is, my understanding is when you talk about biological variations and functionality and differentiation, every gene has a very narrow bandwidth of function. But when you list down your genes of priority, I'm assuming you're doing it based on the numerical levels based on which you see the expression level. So you end up missing genes which are changing very less, but in a biologically more relevant.
Actually, not exactly. We have taken the highly upregulated or highly downregulated sets within our system. It is not what we have done. What we have done is first we have devised the relationship between the microRNA and the target genes, and then we have done a very rigorous literature analysis of how they really change within these particular biological systems. So I guess we have not mistyped those because we have screened the genes not based on only the very top highly upregulated and highly downregulated set. It is the functions on which we have actually emphasized.
Okay. And my second question is the experiments where you see that the clustering of primary cells is always different from your iPSC differentiated cells. You've looked for a set of genes which are contributing to this cancerous phenotype, but did you also identify the set of genes which are unique and are responsible for the primary character of the primary tissues?
Actually, when we did this clustering, that gives us a clue which is actually making them different from the primary count.
So did you see a gene set or an mRNA-RNA interaction which is unique only to, say, primary cardiomyocytes?
The way we started here is a bit different. So first of all, we wanted to fish out those genes, based on this gene expression clustering, those genes which are actually making it different from the primary counterpart. And then we figured out the microRNA mRNA interactions. That is possible, definitely, because what we have done is that will include that thing also, because the primary, the genes which are contributing for the primary cells, those should be there, but to a lesser extent, and that is why, the things is getting deviated. That definitely can be looked upon.
Thank you.
So I had a question here. Yeah. So good to hear about the careful workflows that you have designed. So my question is about the last step in that workflow, in the sense that you use this probabilities to make the calls, right? For those samples that are close to 0.5 probability, should we be treating them a bit more special or giving that information?
Definitely more special, biologically special, I would say, because this is a very spontaneous process, as I mentioned, that when you are differentiating an iPSC, then the cells don't differentiate in the same manner. So might be when you are taking a population and extracting the RNA, part of it do has a pluripotency nature which is actually contributing here. So that is where this thing, and again passaging of the cells, looking at passage number, those are also contributing. So maybe if you keep the cells for a few days, that might shift towards the other side.
I see. So there may be some kind of stochastic decision the cells are making on whether to commit to a cancerous path or a regenerative path.
It's very interesting to see.
Thanks.